In this video, we will discuss about the overexpression of fibronectin in the HFD-fed mice. So, since fibronectins, from their RNA-seq analysis, they are able to find out that fibronectin is overexpressed. So, their focus is mainly on the fibronectins, and they wanted to check whether there is a change in the fibronectin expressions on the endothelial cells when they come in contact with the platelets from the control mice or the HFD diet fed mice. HFD fed mice. So when they check the fibronectins using the staining of these fibronectins on the endothelial cells, they are able to see that in the control mice, the fibronectins for expressions are less compared to the HFD mice. And when they check its interactions with the tumor cells using CD41, they are able to see that CD41 interactions is also increased in the HFD fed mice compared to the control mice. And these things, the F1 expressions are also quantifications of the F1 expressions are shown in this panel K, where we can see that the expressions of the fibronectins are significantly higher in the HFD fed mice compared to the control mice. And since fibronectins is known to trigger intercellular signalings, they wanted to check whether there is a downstream signalings in the fibronectins occurred or not. In order to check that, they target this signaling pathways using SRC and tallying. And what they are able to find out they checked it in the HUVEC cells that there is activations of the SRC which is indicated by phosphorylation of the SRC, and there is also activations of this tallying which is indicated by phosphorylation of the tallying. So this indicates that there is activations in the HFD fed mice in the endothelial cells, which is shown in this in vitro experiments. And now since they wanted to confirm whether the interactions whether this tumor cells interactions with the endothelial cells depend on the fibronectins or not, in order to do that, they use these neutralizing antibodies against fibronectins and check for the HFD mice conditions whether it will reduce the interactions of the tumor cells to the fibronectins. And this is the immunofluorescence image in the panel M, where we can see that in the case of this neutralizing antibodies against fibronectins, we see less tumor cells that are attached to the endothelial cells compared to the conditions where this no neutralizing antibodies are used and the platelets from the HFD mice is used. And these things they have quantified in the panel N, where we can see that tumors with green fluorescence are significantly reduced in the case where neutralizing fibronectin antibodies is used compared to the case where no neutralizing antibodies against fibronectins is used. Next, they wanted to check the cell homing, whether this cell homing in the case of this tumor cells, this integrin alpha 5 plays an important role towards metastasis of the tumor cells. So they wanted to check whether this integrin alpha 5 also has those roles in these conditions or not. And so they checked this highly metastatic mice models, highly metastatic tumor cells, that is E0771, and they use the neutralizing antibodies against integrin alpha 5 and then check the cell homing. So in the case where there is no neutralizing antibodies against integrin alpha 5 is used, they see more cell homings that can be seen in the panel O on the left hand side, the immunofluorescence image. And when they use the integrin neutralizing antibodies against integrin alpha 5, or when they knock out integrin alpha 5 on the highly metastatic tumor cells, they are able to see less cell homings, indicating that this integrin alpha 5 plays important roles towards the cell homings or the metastasis of the tumor cells. And these things, this radiant efficiency is been quantified in the panel P, where we can see that when they neutralize these receptors on the tumor cells, that is integrin alpha 5, they are able to see a decrease in the average radiant efficiencies compared to the case where there is no neutralizing antibodies against integrin alpha 5 used. So from these figures they made 2 points, that the fibronectins is important for the interactions of the endothelial cells and these tumor cells. At the same time, this integrin alpha 5 on the tumor cells are also important for their adhesion properties or the cell homing uh, efficiencies. So I hope you understood that these two findings, how they come up to these two findings. If you need to understand more about these experiments and their final outcomes in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.